Hi everyone, welcome back to my YouTube channel and today we are going to talk about development of dental pulp. So let's start our topic with introduction. Dental pulp is a specialized connective tissue located in the center of tooth, enclosed by dentine and enamel in the crown and by cementum in the root. It contains nerves, blood vessels, lymphatics and various cell types including odontoblasts, fibroblasts and immune cells. The development of dental pulp is closely linked to the formation of tooth itself during odontogenesis. Understanding its development is essential for fields like endodontics, pulp biology, and regenerative dentistry. Definition: dental pulp is soft connective tissue found in the pulp chamber and root canals of teeth, responsible for the formation of dentine, nutrition, sensory perception, and defense against pathogens. Embryonic origin of dental pulp. Dental pulp originates from ectomesenchyme, which is derived from neural crest cells. During tooth development, interaction between oral epithelium, that is ectoderm, and underlying ectomesenchyme guide the differentiation of cells that form the pulp, dentine, and surrounding tissues. Stages of pulp development. Development of dental pulp occur alongside the stages of tooth development that are called as odontogenesis. These stages include initiation stage, birth stage, cap stage, bulb stage, and fifth stage, that is apposition and maturation stage. First stage of pulp development is initiation stage. It starts around the 6th week of embryonic life. In this, the dental lamina forms a thickening of the oral epithelium. Ectomesenchymal cell condense beneath the dental lamina, which will later give rise to the pulp tissue. Second stage is birth stage. It occurs around the 8th week of tooth development. Dental lamina proliferates into buds that penetrate the ectomesenchyme, and these ectomesenchymal cells start differentiating into pulp progenitor cells. Third stage is cap stage. It occurs around the 9th to 10th week of development, and during this, the enamel organ forms a cap over the condensed ectomesenchyme. The condensed ectomesenchyme beneath the enamel organ is called the dental papilla, which will form the dental pulp and dentine. Outer cells of dental papilla differentiate into odontoblasts, which will produce dentine. Fourth stage is bulb stage, that is around the 11th to 12th week of embryogenesis, and during this dental papilla further differentiate into peripheral cells and central cells. Peripheral cells form odontoblasts that will produce dentine, and central cells form fibroblasts and undifferentiating mesenchymal cells that will form the main pulp tissue. Pulp develops its vascular network, nerve fibers, and primitive connective tissue. Fifth stage is apposition and maturation stage. Odontoblasts secrete dentine, which later mineralize into dentine. The pulp cavity form containing connective tissue, blood vessels, and nerves. Cementoblasts forming from the dental follicles surround the root portion. And during this, the pulp mature as tooth eruption approaches, acquiring full vascularization and innervation. Cellular components of dental pulp include odontoblasts that form dentine along the periphery of pulp they are present. Second cellular component is fibroblasts that produce collagen and extracellular matrix. Third component is undifferentiated mesenchymal cells that serve as the progenitor cells for repair and regeneration. Fourth cellular component is immune cells that provide defense against infection. Fifth component is nerve fibers that begins entering the pulp during the birth stage to provide sensory function. Functional significance of dental pulp development. First is dentine formation, as odontoblasts produce primary, secondary, and tertiary dentine. Second is nutrition, as blood vessels supply oxygen and nutrients to the tooth. Third is sensory functions, as nerve fiber allow perception of pain and temperature changes. Fourth is defense, as immune cells protect against bacterial invasion and injury. Fifth is repair and regeneration, as undifferentiated mesenchymal cells differentiate into odontoblasts-like cells when pulp is injured. Factors affecting development of dental pulp. They include genetic factors. They control cell differentiation and tissue patterning. Second factor is signaling molecules like BMP, FGF, WNT, and SHH pathways that regulate odontoblast differentiation. Third factor is nutrition and maternal health, as proper maternal nutrition and absence of teratogens support normal pulp development. Fourth factor is environmental factor, as trauma, infection, or chemical exposure during tooth development can disrupt pulp formation. Conclusion of this lecture video. Development of dental pulp is a complex, highly coordinated process that begins early in tooth formation and continues as tooth matures. It originates from neural crest-derived mesenchyme and differentiate into odontoblasts, fibroblasts, and connective tissue cells. Proper development of pulp is essential for dentinogenesis, tooth vitality, sensory perception, and defense mechanism. Understanding pulp development provides a foundation for clinical practice in endodontics, pediatric dentistry, and regenerative dental therapies. And this was all about dental pulp formation. Hopefully, you liked the video. If you do, don't forget to like, share, subscribe, and comment. Thank you for watching.